Welcome back. This lesson is on monohybrid cross and the learning goals to be achieved from this session are 1. Know and use the genetic terminology of Mendelian inheritance. 2. Explain the steps of Mendelian cross. 3. Illustrate monohybrid crosses up to F2 generation. 4. Recognize the dominant and recessive traits or alleles in the monohybrid cross. 5. Calculate the genotypic and phenotypic ratios of F2 generation. 6. State the two laws formulated by Mendel for the inheritance of one trait. And 7. Determine unknown genotypes by test cross. In the previous lesson, we learned that Mendel conducted two types of hybridization experiments which involved crossing of two true breeding pea plants that either differed in one trait or two traits. And we also realized that in the naturally self-pollinating pea plants, Mendel was able to achieve this through controlled pollination, where he manually transferred the pollens from the desired male parent with one variety to that of the stigma of a female plant with the other variety for the same trait. Now, before I go into my actual lesson on monohybrid cross, I would like to introduce you to some of the genetic terminologies which forms the essence for understanding Mendelian crosses. The first two terms are a kind of a recap from the previous lesson, where the first term here is monohybrid cross. Now this, as we learned earlier, refers to a cross between two parent plants that differ in one trait. And to give you an example for such a cross between tall and dwarf pea plant is an example for monohybrid cross, because in this cross, the two parent plants differ in stem height. The second term is dihybrid cross, and dihybrid cross refers to a cross between two parent plants that differ in two traits. For example, a cross between round and yellow seeded plant with a wrinkled and green seeded plant is an example for dihybrid cross, because here the two parent plants are differing in two traits, which are seed shape and seed color. The third term is gene. Gene can be defined as the basic unit of heredity that passes from the parents to the offspring. Or in another way, we can also define gene as a segment of DNA that determines a particular trait in an organism. For example, BRCA1 and BRCA2 genes of humans. Now these two genes I have listed in one of our previous lessons pertaining with breast cancer. Next term is allele. Allele refers to the different forms of a gene. And in most of the organisms, genes have two alleles. Right? And these alleles are to be represented with letters when we are making genetic crosses. For example, the two alleles of stem height in pea plant are denoted by capital T and small t. So capital T is used for tall trait and small t is used for dwarf trait. Now in this context, allele can be further differentiated into two types of alleles. It can be a dominant allele or it can be a recessive allele. Dominant allele refers to an allele that can express even in the presence of the other allele. So we know that each gene has two alleles. So if the two different alleles are present together in an organism, in such a situation, dominant alleles are the ones which are able to express even in the presence of the other allele. And these are basically represented with a capital letter. For example, in capital T small t combination. So here is two of the alleles, two different alleles existing together. So in such capital T small t combination, capital T expresses itself and masks or hides the character or the trait of small t. So capital T, because it's able to express its character, that is, the plant are going to be now having tall trait, and the other allele, which is the small t, is masked by the presence of capital T. So capital T or the tall allele becomes the dominant allele. The second type of allele is the recessive allele. So a recessive allele refers to the allele which gets masked or hidden in the presence of the dominant allele. So if we have to, and these recessive alleles are represented by lowercase letter, okay, small letters. And if we take the same example, that is in the capital T small t combination. So small t is the character of the small t. That is the dwarfness of the small t. The dwarf character of the small t is masked by the presence of capital T. So a small t cannot be making its expression in the presence of capital T. And because of which, because it's getting hidden here, it's known as recessive allele. Some of the terms that are to be frequently used in Mendelian crosses are genotype, phenotype, homozygous and heterozygous. The term genotype refers to the genetic makeup or constitution of an organism, or it can also be defined as the collection of alleles in an organism. For example, if we consider the stem height trait in pea plants, so the tall height is denoted by a genotype which can be capital T, or it can be a capital T small t genotype. Whereas the dwarf height trait of the pea plant is denoted by small t genotype. So you can observe here that a genotype is composed of two alleles. The term phenotype refers to the observable physical characteristics of an organism, which is to say we are talking about the external feature of an organism. And this is expressed in genetic crosses in words. For example, the stem height trait is expressed as tall and dwarf. Now, the two terms homozygous and heterozygous are directly related with genotype. So, homozygous refers to a genotype with two identical alleles for a trait. So, if we have a genotype which is composed of two identical alleles, such genotypes are referred to as homozygous genotype. For example, capital T genotype for tall height, right, is an example for homozygous genotype because both of the alleles are capital T's. The second example is the genotype which is for dwarf plant, which is small t. So both of the alleles are small t's. Whereas a heterozygous genotype refers to a genotype with two different alleles for a trait. So now in this genotype, the trait is carrying two different alleles. For example, capital T small t genotype. So this is a genotype even for tall plants, but then in this genotype you can see two different alleles. One capital T and the other one is small t. And such kind of genotypes are heterozygous genotype. The last three terms that I'm going to define are true breeding lines, parental generation and filial generation. True breeding lines refers to all the organisms that always passes down its own trait without any variation to the offsprings from generation to generation. And the reason why these true breeding lines are capable of maintaining with the purity of their character is because they possess homozygous genotype. For example, a homozygous tall parent plant with a genotype capital T will always be producing offsprings that also show the tall character. And such kind of true breeding lines are always used by Mendel for his different crosses. Parental generation refers to the organisms that are to be used as parents for a cross. So it refers to the plants used as parents in a cross, and parental generation is designated by the letter capital P. Last, we have filial generation. Filial generation refers to the progeny of a particular generation. So the progeny obtained as a result of genetic crosses are known as filial generation. And to give you two specific examples, progeny obtained from the parental generation are known as F1 generation or the first filial generation, and the progeny which are obtained from the F1 generation are known as F2 generation, that is the second filial generation. So these are the technical terms that you have to be very clear before we try to move ahead with the understanding about all different kinds of Mendelian crosses and genetic crosses. Well, let's now proceed to make sense of Mendel's monohybrid cross. The purpose of Mendel's monohybrid cross was to study the inheritance of one trait, or in today's language we can say it was to study the inheritance of one gene. One such monohybrid cross was a cross between tall and dwarf pea plants. So we will try to study the detail of Mendelian monohybrid cross by taking this as an example. So in the first step, what Mendel did was Mendel selected true breeding tall and true breeding dwarf plant for his parental generation. So he had P generation, or the parental generation, where he crossed a true breeding tall plant to a true breeding dwarf plant. And in this example, let's say that Mendel chose tall plant as his female parent and made the dwarf plant as his male parent. So in the second step, what Mendel did was Mendel performed controlled pollination, which is to say he dusted the pollens from the dwarf pea plant over to the stigma of the female tall plant. So in this way, he was able to cause cross pollination as well as cross fertilization. Now, after obtaining the seeds from such crosses, Mendel grew the seeds in order to get his the first filial generation, or the first progeny generation. So now he got the first filial generation, or F1 generation. And I must emphasize here that Mendel based his observation purely on the phenotypes of the offsprings. So what Mendel did was Mendel tried to study the phenotype of each of his F1 plants. And what he noticed was all of his F1 plants just showed only one phenotype. They all showed tall-heighted phenotype. Okay, so now in order to confirm with his result, what Mendel also did was he made reciprocal crosses. Reciprocal cross means to say basically the reversing of the sexes of the parents. So he performed another cross in which now he had chosen in this cross dwarf as the female parent and tall as the male parent. And again he had dusted the pollen from the tall male parent over to the stigma of the dwarf female parent, and in that way he was able to cause cross pollination and cross fertilization. And when he obtained his first filial F1 generation and observed the phenotype of all of his F1 plants, he was again having all the progeny showing only the tall character. So what Mendel noticed here was that in his monohybrid cross between the tall and dwarf pea plants, he always obtained F1 plants which showed the tall phenotype. Now this intrigued Mendel because he did not find any kind of a blending or a mix of the parental characters in his F1 plants. He did not have intermediate height in the F1 plants, which was actually the widely accepted theory during on inheritance during his time. Right? So during his time there was this popular theory called as blending inheritance theory. So he was not able to find any kind of a blend of the parental characters, but rather he saw that all of his F1 plants carry just one parental traits and the other parental trait was completely absent. So Mendel wondered what happened to the character what happened to the dwarf character in the F1 generation. To figure out the mystery, Mendel went ahead with his study on the subsequent F2, F3 and more generations. And all of these subsequent generations were obtained through continuous self-pollination. So, in order to obtain the F2 generation, he made the F1 plants to undergo self-pollination. And this step is F1 selfing, where F1 plants are to be allowed to undergo self-pollination, which basically means to say that the pollination would happen within the same plant. And he then collected the seeds of such F1 self-pollinated plants and grew them in order to get the F2 generation or the second filial generation. Now, when Mendel made his observation on each of the F2 plants, what he noticed was that he had both of the parental phenotype in the F2 plants. So, F2 plants showed both parental phenotype. He could be finding the dwarf phenotype reappearing in the F2 generation. And as I said in my previous lesson, what is unique to Mendel's experimental work is not only did he observe the phenotypes of the progeny of each of the successive generations, but he also counted each and every plant with a particular trait. So in this experiment, he actually obtained 1064 of F2 plants, and out of the 1064 of the F2 total plants, he noticed that 787 plants were showing the tall character, whereas the remainder 277 of his F2 plants had dwarf height. So this was the exact number that he counted of his F2 generation, where 787 had the tall character and 277 had the dwarf character. And what Mendel noticed here is that 787 is to 277 is almost equal to a 3:1 ratio. So this was the phenotypic ratio that he obtained in F2 generation. So F2 phenotypic ratio was almost 3:1, where of the total plants, 3 part of the plants had the tall character and 1 part of the plant had dwarf character. Or in another way we can say 75% of the plants showed the tall character and 25% of the plants had the dwarf character. Mendel confirmed his result by repeating the crosses for the 6 other pairs of contrasting traits in pea plants. In each case, what Mendel observed was that one of the parental character disappeared in the F1 generation and it reappeared in 1/4 of the F2 generation. In all of the several thousands of monohybrid crosses that he conducted, the average phenotypic ratio in F2 was 3:1. So basing on these experimental results, Mendel finally proposed 2 major laws in order to explain for the inheritance of one trait. Some of the inferences or conclusions that Mendel made based on his monohybrid experimental results are the following. 1. That traits are controlled by discrete units called factors. And second, that these factors exist in pairs. So Mendel said that all the traits are controlled by factors and that these factors are always to be occurring in pairs. Today, what we know is that what Mendel was proposing as factors are the genes, and the pair of factors is referring to the alleles of the gene. Apart from this, Mendel also proposed two laws of inheritance. The first one is law of segregation. Now, this law states that the pair of factors segregate from each other during gamete formation. So, as we know, Mendel suggested that the factors are to be existing in a pair, and such pair of factors should always be segregating or separating from each other during the time of gamete formation, and each gamete would only be receiving one of the two factors. So this law is called as law of segregation. And the second law that he also proposed was law of dominance. This law states that in an organism with dissimilar pair of factors, so if an organism has two dissimilar alleles for a particular trait, or if an organism has a heterozygous genotype, right? Organisms with heterozygous genotype are known as heterozygote. So the condition is heterozygous, and the individual is known as heterozygote. So an individual with heterozygous genotype, one factor which expresses and masks the other factor. So of the two dissimilar allele, one allele would be making its expression, and at the same time it's going to be masking the expression of the other allele. So this allele which makes its expression and which masks the expression of the other allele is known as dominant factor, and the factor which is masked by the dominant allele, this is called as the recessive factor. So basically dominant factor and recessive factor are again referring to the dominant allele and recessive allele. So this law is referred to as law of dominance. And what we know is Mendel's factor or Mendel's traits are always to be showing a complete dominance and a complete recessiveness. So we know that Mendel selected 7 pairs of contrasting traits in pea plants for his study. Now, from the several thousands of the monohybrid crosses that he conducted, he finally determined or confirmed with the dominant and recessive traits in each of these 7 pairs of contrasting traits. And I would say that you need to know all of these dominant and recessive traits for the 7 traits in order to be able to work on Mendelian crosses. So this is how it is. For stem height, tall is dominant over dwarf. And the dominant and recessive traits are to be expressed in letters. And as we know, for the dominant trait, it has to be a capital letter, and for the recessive trait, we are going to be using the same letter, but it has to be lowercase letter. So, stem height, tall, denoted with capital T, dwarf, denoted with small t. Likewise, for seed color, yellow color is dominant over green. So, we are going to be using capital Y for yellow and small y for green. For seed shape, round shape is dominant over wrinkle shape, and the letter that we are going to be using here is capital R for round and small r for wrinkled. Pot color is, if you just try to see with the seed color and the pot color, you can see a kind of a reversal. So for pot color, green color pot is dominant over yellow color pot. So the letter that we are going to be using here is capital G for green and small g for yellow, because yellow pot is the recessive trait. For pot shape, inflated is dominant over constricted, and the letter that we are going to be using is capital I for inflated and small I for constricted. In flower color, purple or violet color is dominant over white color. So if you're going to be referring to the flower color as purple color, then we are going to be using capital P, or sometimes we even have the usage of violet color for the dominant color. And if you're going to be referring violet as the dominant color, then we are going to be using capital V for violet. And for white, basing on which dominant color you are using. If you're using purple, then white would be denoted with a small p. If you're using violet, then you're going to be using small v as the denotation for white color. And finally, flower position, axial is dominant over terminal. So axial is denoted with capital A and terminal is denoted with small e. So these are the 7 traits with all of their contrasting dominant and recessive traits. Let me now work out one of the monohybrid cross between tall and dwarf pea plants. And I want you to pay extra attention to the format of this cross because this is going to be remaining same for all of the Mendelian crosses. Now the first step that we need to do here is to assign letters for the 2 characters. And from the experiment, Mendel learned that tall was dominant over dwarf. So we can assign, let tall be capital T because it was dominant, and let dwarf be small t. After assigning the letters, then we are to show the parental generation, which is P generation. Now, the P generation has to be shown both in the phenotype as well as genotype. So, we are first going to be writing the phenotype of the 2 parents. So, we have a cross between true breeding tall and a true breeding dwarf plant. And you can be even assigning the sexes for the parents, right? And now we need to assign the genotype for tall and dwarf. And since we know that Mendel's parents are always true breeding, and because we know that tall is the dominant character, so hence genotype of tall would be 2 capital T's and the genotype of dwarf would be 2 small t's. Okay, so with this we have finished with the P generation or the parental generation. Now in the next step, we are to show with the formation of gametes from the male and female parents. And as proposed by Mendel in his very important law, which is the law of segregation, so the pair of factors, right, the pair of factors in the organism are to segregate from each other during the time of gamete formation. So this is the step that we want to show in gamete formation. So during gamete formation, you have to show that there is segregation of the pair of factors. And this results in 2 possible gametes from the tall female plant, which can be basically they can be producing 2 gametes, but both of the gametes are going to be identical because the genotype was homozygous. Likewise, again we have the segregation of the 2 small t's and they are going to be going independently into each of the male gametes. So we have 2 possible male gametes, but the male gametes are basically having the same allele because it was a homozygous parent. Okay, so this is how we denote with the law of segregation or gamete formation from the 2 respective parents. The next step here is now we have to show the F1 or the first filial generation, and to show the possible combinations of fertilization or to show with the possible combinations of genotypes in the F1, what Mendel did was Mendel used a mathematical tool known as Punnett square. So to show with the possible combinations in the F1 generation, Mendel used Punnett square. So Punnett square basically is a grid that we are going to be drawing, or a box, the square boxes that we are going to be drawing. And the number of square boxes that we need to draw depends on the number of gametes from the parents. So here in this case, we have 2 gametes from the female parent and 2 gametes from the male parent, and hence we need 4 boxes. So let's show the 4 boxes by drawing 2 columns and 2 rows. After drawing the box, we now have to place the gametes on the grid. And one of the parental gametes are to be placed on top of the grid, and the other gamete are to be placed on the left side of the grid. So here what I've done is I placed the 2 male gametes on the top of the grid, and the other 2 female gametes on the left side of the grid. Now it becomes easy for us. Inside the box, we want to be filling with the possible combinations of genotype for the particular offspring. So in the first possible combination, what we have to do is we have to take the first female gamete and the first male gamete, and this would be resulting in a capital T small t combination of genotype. Then the second possible combination is the first female gamete T with the second male gamete small t, and again it's going to be capital T small t. So basically we are going to be having same kind of combinations here, because both of the alleles of the male and female gametes are the same. So here again we have capital T small t, capital T small t combination. So in this way we have completed the 4 possible genotypic combinations for the F1 offspring. So the use of Punnett square is that it works on the principle of probability, and thereby we are able to be predicting with the possible genotypes for an offspring from a genetic cross. So now from this box we are able to study the genotype of the F1 plants. So if you look into the box, all of the genotypes are the same, right? So 100% of the genotype are all capital T small t heterozygous. So this is the heterozygous genotype, which you see in all the F1 plants. And if you consider the phenotype of the F1 plants, all of these F1 plants with such kind of a genotype was all tall. Okay, so we know that capital T is dominant over small t, and because of which in the presence of capital T, small t cannot be making its expression, and hence all of the F1 phenotype was tall phenotype. After this, the next step is F1 selfing. And F1 selfing is denoted by a cross between heterozygous genotype, capital T small t with that of a capital T small t. Now we need to remember here that these two are not indicative of 2 separate parents, because this is purely a self-pollination. It's happening within the same plant, but these are a denotion for, you can say, the sex cells of the plant. So within the same F1 plant, female sex cells are to be producing female gametes, and the male sex cells are to be producing the male gametes. So these are referring to the sex cells. And now again we will follow the same format. From these the parental cells, we expect the formation of gamete. So we will show the gamete formation step, and again we have to apply law of segregation here, which means to say the pair of factors which are present in each of these parental sex cells are going to be segregating from each other at the time of gamete formation, and each of the sex gametes, or the egg and the male gamete, are going to be receiving one of the two factors. So let's show this. So in the female, the sex cell factors are going to be segregating into two types of egg, where one egg will be having capital T as the allele or the factor, and the other egg would be having small t as its allele or factor. Then likewise, the male sex cells are also going to be producing gametes, and the gametes are always going to be receiving just one of the two factors. So one of the male gamete would receive capital T, the other male gamete would receive small t. So what you see here is, when the genotype is heterozygous, then we have even two different types of gametes produced. When the genotype was homozygous, at that time we always had the production of same gametes. But the number of gametes remain the same. We have two gametes produced by the female sex cell and two gametes produced by the male sex cell. Now these gametes are to be fusing and then resulting in the formation of subsequent generation, which is the F2 or second filial generation. And again, in order to show all the possible combinations of genotypes in the F2 plant, what Mendel did was Mendel applied Punnett square. So we have to show the Punnett square, and in order to decide with the number of boxes for the Punnett square, we need to be looking into the number of gametes. So we have two gametes, each from the male and female sex cell. Thereby we are going to be again drawing two columns and two rows. Okay, and then we have to place the gametes, one on top of the grid and the other parental gamete on the left side of the grid. So let me place this male gamete. So these two, I'm going to be placing it over here, one on each or the top of the column, and then the female gametes, I'm going to be placing it down here. Okay, so one on each of the left side of the row. And now what we need to be doing is, as we did before, we want to be having the combination made for each of these boxes. So the first box would be having a combination of first female gamete and the first male gamete, and here the first female gamete has a capital T, the first male gamete has a capital T. So hence the genotype becomes capital T, capital T. Now in the second box, the genotype is going to be becoming the combination of first female capital T egg and the second male small t male gamete or the sperm. Okay, then in this row we have the first female gamete, which is the second female gamete, which is small t, and the first male gamete, which is capital T. Now when we have to write this down, we always need to write the capital letter before the small letter. Okay, so instead of writing small t capital T, we are going to be writing capital T small t. Okay, so we write the capital letter always before the small letter, and the last box would be having a combination which is small t of the female gamete, and the second male gamete is also small t. So we will have two small t genotype. So these are the four possible genotypes of F2 generation. Now, if you look into the combinations of the genotype, unlike in the F1 plants, we can be finding that the boxes have some kind of differences in their genotype. So let us now write down the F2 genotype. But F2 genotype is not the same for all the plants. Some of the plants can be having a genotype which is homozygous capital T, capital T. Then another type of genotype that we see here is in this box as well as this box, where two of the boxes are having heterozygous genotype, which is capital T, small t. And the last box here has another type of genotype, which is homozygous recessive genotype, which is small t, small t. So from such kind of a Punnett box use, we are able to predict three of the possible genotypes for F2 plants. It can be capital T homozygous dominant, it can be heterozygous, or it can be homozygous recessive. So this is homozygous dominant. Both alleles are dominant. This is heterozygous. And the last one is homozygous recessive. So these are the three types of genotypes in F2 generation. And Punnett square can also be used in order to predict with the frequency or the ratio of the genotype. Okay, so let's write F2 genotypic ratio. So what is the F2 genotypic ratio? Here, out of the four possible genotypes, one box has homozygous dominant genotype. Okay, so 1/4 of the progeny can be having a homozygous dominant genotype, and 2/4 of the progeny can be having a heterozygous genotype. Okay, because two boxes are having the heterozygous genotype. So 2/4 of the progeny can be having heterozygous genotype, and one box has the homozygous recessive genotype, which is 1/4 of F2 progeny can be having homozygous recessive genotype. So now the genotypic ratio for the F2 generation becomes 1:2:1. Okay, which is very consistent and very typical for Mendelian monohybrid crosses. Then we can also talk about the phenotype of F2 plants. So what can be the phenotypes of the F2 plants? So by looking at the Punnett square again, we can be predicting with the possible phenotypes. So since we know that T or tall is dominant over small t, which is dwarf, so three of these genotypes, the homozygous dominant and heterozygous genotype, possessing plants can be expressing tall phenotype, whereas organisms with this homozygous recessive kind of genotype can be expressing dwarf. So as Mendel also noted in his the numbers of the tall and the dwarf F2 plants, so we can also be seeing through the Punnett square use that out of the four possible you can say genotypes, three of the genotypes can be resulting in a tall phenotype, and one of the genotype can be resulting in the dwarf phenotype. So this would be basically giving us the ratio of phenotype in F2. So let's say this is the phenotypic ratio. So the phenotypic ratio in F2 is 3:1. Okay, so 3/4 of the F2 plants can be having a tall phenotype, and 1/4 of the F2 can be having a dwarf phenotype. Right, and this was a consistent and a very typical result of all of the Mendelian monohybrid crosses. So with this, we have completed with the monohybrid cross between tall and dwarf pea plants up to F2 generation. Finally, let's do some more study into the cross. So we learned that tall phenotype is the dominant phenotype over dwarf. And tall phenotype is present in the parental generation. It was expressed in the F1 generation as well as it was expressed in the F2 generation. But if you look into the genotype of the tall plants, we can be finding two different types of genotype. Parental genotypes were the homozygous dominant genotype with two capital T's, but the F1 hybrids had the heterozygous dominant genotype with capital T and small t, and in the F2 as well we have some of the tall plants having the homozygous capital T combination of genotype, and the rest of the other tall plants having a heterozygous dominant genotype, capital T, small t. So now how did Mendel confirm, or how did Mendel test that the genotype of a particular dominant trait, like for example the tall trait, is a homozygous genotype or a heterozygous genotype? So for this, Mendel managed to design another type of cross which is known as test cross. Well, what is a test cross? Test cross is a cross which is used to determine whether an organism with a dominant phenotype is homozygous or heterozygous in its genotype. In this cross, the plant with the unknown genotype is always to be crossed with a recessive plant for the same trait. The reason why we select a recessive plant is that we know the genotype of the recessive plant. Recessive plants are to be having homozygous recessive genotype. Now let's take an example. Suppose we want to determine the genotype of a tall plant. We are going to be crossing the tall plant with a dwarf plant having small t homozygous genotype. Now what Mendel proposed was, when we make such test crosses, we are to be expecting two possible results. And I have the two possible results shown here. So, the two possible results are primarily to be based on the phenotype of F1 progeny. Okay, so the phenotype in the F1 or the phenotype in the offsprings become very important in terms of determining with the genotype of the dominant plant. Now let's take one of the possibilities first. So in this case, when we make a cross between a tall and a dwarf plant, if we obtain F1 plants where all of the F1 plants, 100% of the F1 plants are always expressing only one of the phenotypes, that is the dominant tall phenotype. So if we find the F1 plants to be expressing only with the dominant phenotype or the tall phenotype, then we can be inferring that the genotype of the unknown tall plant is a homozygous genotype. So in order to prove this, what Mendel did was, Mendel worked out the cross using Punnett square. So when we say it has to be homozygous, the tall plant is expected to be having two capital T's. So if we have two capital T containing tall plant crossed with a homozygous recessive dwarf plant and make the cross here, so you have four possible genotypes that can be achieved from this cross. Now we know that the four possible genotypes here are capital T, small t, heterozygous genotype. And in such a heterozygous combination, we can never be expecting the expression of the small t or the dwarf character. And as a result of which, all of these progeny or all of this F1 will always be having the tall phenotype because they have heterozygous genotype. And that heterozygous genotype can only be achieved if both of the alleles in the tall plant is capital T and capital T. Now the second possibility that we can be also deriving from such kind of a test cross is that out of the total F1 progeny, 50% of the progeny might be expressing with the dominant phenotype or the tall phenotype, and 50% of the progeny might be expressing with the dwarf phenotype. So if we get such kind of a result where both tall and dwarf phenotypes are expressed in the F1 progeny after making the test cross, then we can be inferring that the genotype of the tall plant is a heterozygous genotype, which is to say it has to be capital T small t genotype. And Mendel again proved this by working out the cross using Punnett square. So here the tall genotype is taken as capital T small t, a heterozygous genotype, and the genotype of the dwarf plant is small t small t. So we are to be having two small t's as the possible gametes from the dwarf plant, and capital T and small t as the two possible gametes from the tall plant. And because we have a small t as a possible gamete from the tall plant, we can be finding that out of the four possible genotypes for the F1, the 50%, or you can say two of the boxes of the possible genotype can be a homozygous recessive small t genotype. And because we know that small t can be expressing when they are in the homozygous combination. So we have 50% of the F1 expressing the dwarf phenotype and 50% of the F1 expressing the tall phenotype. So in this way, whenever we have a 1:1 ratio of the tall and the dwarf phenotype expressed in the test cross, then we can be inferring that the genotype of the tall plant is a heterozygous genotype. So this technique is known as test cross, and by using test cross, Mendel was able to determine, or Mendel was able to predict with all of the genotypes of his F1 plants, F2 plants, and even with the subsequent generation of the dominant plants. So with this, I come to the end of today's lesson, and I'm going to be coming up with my next video on dihybrid crosses.